Good afternoon, everybody. Welcome to our second Thursday seminar. Before I turn it over to Cody for the introduction, I just want to point out a couple of things. Yoda has created several different posters for our photo contest, our seminar series, and a bunch of QR codes. You've probably seen this before about life in Ann Arbor, so grocery stores, outdoor activities, indoor activities, etc. And as you leave this room and turn left, there's a poster right by the door that advertises our Instagram account. So if you want to keep up with all the cool things that are happening in EEB via Instagram, that's the spot to do it. Cody, here you go.
As long as I can catch. Okay. Thank you, everybody, for coming this afternoon. I'd like to introduce you to Dr. Bryan McLean. Bryan received his bachelor's degree from Appalachian State University, which is of U of M fame, if you guys know that. And then attended UNC Wilmington for his master's degree, working under the supervision of Stephen Emsley. Bryan received his PhD at the University of New Mexico with Joe Cook. I think some of you might know Joe. Bryan was also a Peter Buck predoctoral fellow at the Smithsonian. Following his graduate work, Bryan worked as a postdoctoral fellow with the University of Florida with Rob Gerlnick and Dave Blackburn and David Reed. So quite a group. In 2009, Bryan joined the faculty at the University of North Carolina, Greensboro as an assistant professor. And he also maintains an adjunct curatorial appointments with the Museum of Southwestern Biology at the University of Mexico and the University of Florida Museum of Natural History. Bryan's research focuses on landscape to continental scale patterns of mammalian biodiversity and responses of these systems to past and present environmental change. His lab work is data-driven and blends field, laboratory, and biodiversity informatic approaches to understand the captivating global diversity of mammals, how this diversity evolved and is maintained, and how it is responding to current global change. For his work, Bryan has actually already received nearly $5 million in grants and is published in top-tier journals such as Bioscience, Ecography, Ecology, Evolution, and PNAS. Speaking personally, I've known Bryan for several years. We both worked on ground squirrels for our dissertations, and our research interests have overlapped since then. We actually currently have a couple grants together, which I think we'll probably talk about at least one of those today. And with that, please join me in welcoming Bryan to Michigan.
Thanks a lot, Cody, for the introduction. Can everyone hear me? I think so. Great to be here. I really appreciate the invitation. I've really enjoyed talking to some of you. I feel like my world shrank while I was here because everyone I talked to, basically, there was 1 degree of separation between us and right? We were connected by one other person that we had in a mutual friendship. So it's been great to have my world shrink while here and hear about all the great research happening in the department. I want to start today by thinking just about the concept of biodiversity data. What are those? Who generates them and who needs them and who uses them? This is an image that might be familiar to some of you. This is from the United Nations Convention on Biological Diversity COP15 meeting that took place in 2022, if you were paying attention then. If you're not, there's a meeting called COP16 coming up this year in Colombia. This is basically a venue for world representatives to get together, talk about biodiversity conservation, and establish and assess progress towards targets for conserving biodiversity. This is an example of who needs biodiversity data. That might be the highest level at which biodiversity data are used and kind of summarized for policy. But the flow of knowledge from primary observations to that point, which will be right here, is nuanced, right, to say the least. At some point, primary observations of biodiversity are usually generated by people like you going out in the field, detecting genes or species or populations out in the landscape and recording that, or maybe passive sensors. And these can be summarized or synthesized into multiple different products. In this case, what have been called essential biodiversity variables. And then these inform basically higher level change indicators that people can use for policy and monitoring. Okay, so these ideas of essential biodiversity variables have kind of evolved into a set of about 6 classes of variables. And I think this is a pretty decent way of thinking about biodiversity knowledge, how we summarize it and use it for research and conservation. These have been discussed quite a bit in the literature recently, so you might have heard of these or read papers trying to flesh out what one of these classes is and how we define variables within it. But basically, they encompass levels of organization. They range from genes to species to communities and ecosystem, both structure and function. And then within each of these classes, there's a set of variables that we can apply to different species in different regions and assess biodiversity trends. It would be nice if all of these could be tracked with equal effort, right? If we had data for all of them, if we could go to each level and assess response to environmental change at each level with the same level of detail. But while these concentric rings are all the same, what is actually, the reality is much more like this. This is an image from the Europe group thinking about this. Data on species occurrences and populations are usually these big sections of the circle. There's substantially fewer candidate variables and fewer information for things like genetic composition and species traits. And so information about the genotypic and phenotypic properties of species is pretty rare out there, and we don't have much tractability when we try to monitor them at scale. This is unfortunate because traits interact with many aspects of an organism's environment, right? And by traits, I mean morphological, physiological, life history, and other properties of species. And these are the types of ways that traits interact, and they mediate a lot of different outcomes, right? Trait by environment interactions can mediate things like individual fitness, the persistence of populations, and even the evolutionary success of lineages. One point I hope to get across today is that my lab is thinking a lot about these traits at the individual level. And this is really the level at which these interactions are occurring, right? It's the traits of individuals and populations that experience these interactions and by which these outcomes happen. And it's essential that the trait data we have available to us are at that same level. But as you might have guessed from what I've said so far, these trait data are pretty sparse and fragmented across space, through time, and among taxa. So it's not so much that we can't think of questions we could ask with traits. There's a variety of traits that could inform us about biodiversity response, ecology, and evolution, just like the variety of materials and ingredients available to this chef. But this is what we've been given. A few traits that are very difficult to scale up. And if you ever watch this show, you get one ingredient to make a complex dish around. So this is kind of what we have when it comes to traits. We have a few traits that it's very difficult to scale up and ask questions at bigger levels with. So today I want to talk about what very simple traits of mammals might tell us about ecology, physiology, and possibly global change response in mammals. The first two projects are related to plasticity, and I've talked to some of you today that I met with a lot about these ideas. Specifically, I will talk about some work thinking about plasticity in brain and body size. This is in populations of mammals across seasons. And also some plasticity work in the GI tract, same. Within species work across seasons, thinking about how these traits and changes in them and plasticity in them facilitates survival. And then I will talk a little bit, as Cody said, about how we're scaling up this trait-based work to capture more trait data from museums and how institutions like this are playing a role in that. So let me get started without skipping ahead too much. Okay, so mammalian energetics is a really rich field. It's historically been considered to be a balance in a very basic equation between incoming resources, maybe modulated by environmental parameters like the temperature or the photoperiod, So a balance between the incoming and then the critical tasks, right, of reproducing and surviving and growing and maintaining tissues. So this is kind of the basic equation when it comes to energetics, and this is a model that was developed for rodents. But to maintain energetic balance in seasonal regions, survival strategies, energy-saving strategies are really key, especially in temperate mammals, and that's a system I work in a lot. So in mammals, there's a variety of energy-seeking strategies. These can be things like adjusting home range, dispersing, migrating, undergoing some kind of behavior change. And there's a variety of energy-saving strategies, although fewer, right? Things like using daily torpor or hibernation to modulate your energy needs. What if you could just reduce your total energy demand? That seems like the easiest way to do this, right? Reduce the amount of energy you need, and you don't have to do any of these other things. So I'm going to talk today about this phenomenon that's been called Daynell's phenomenon. And Daynell's phenomenon describes a seasonal and anticipatory decrease in body mass, brain case and brain size, and some other structural proportions, and working out how that's manifested. But this change is occurring from summer to winter, and it's followed by a regrowth in spring. So it's kind of plasticity in the classic sense. And this is a photo of the species in which this phenomenon has been investigated the most, Sorex araneus, the common shrew, distributed across Europe and Asia. And these are just some x-rays, in this case, of the lateral view of the cranium, showing progression of age classes. So this is a first summer and a first winter and a second summer. And the phenomenon has been described a lot in the proportions of the brain case. So this is involving a flattening of the brain case and then a regrowth in the second summer. But this is manifested also in body mass and things like that. This works as a strategy, in theory at least, because as most of you know, metabolic rates scale strongly with body size in mammals. And so reducing your total body mass should lead to an overall reduction in the amount of energy you need. Much of Daynell's phenomenon, as I said, is focused on this crazy phenomenon of brain case change and therefore brain shrinkage, which has also been studied. And this also makes sense as one place to focus because the brain consumes a lot of energy. So for example, in humans, it consumes, or it makes up about 2% of your body mass, but uses about 20% of the energy. This makes sense as a place where you might save some energy if possible. But there are some other tissues, organs, and organ systems that also use a lot of energy. For example, skeletal muscle takes up about 42% of your body mass on average. It doesn't use a disproportionate amount of energy, but it uses a large amount of energy for the total mass that it takes up. So general body size reductions, muscle reductions could be another place to save energy. But we don't know much about how this manifests across other parts of the phenotype, right? We know something about body mass, something about brain case proportions, and maybe brain size, but we don't know how this is manifesting across the entire phenotype. In fact, much of our knowledge of this phenomenon is pretty uneven. As we dug into this, there's some major geographic and taxonomic biases in the data. About 50% of the studies we found, and I'll talk about that in a minute, were on one species, the species I showed you, Sorex araneus, which is distributed across a lot of Europe and Asia. So about 50% of the knowledge is from one species. There's 88 species in this genus. There's limited documentation of the phenomenon in North America and East Asia. And then there's been no documentation of plasticity in both the body mass and the skeletal proportions below about 43 degrees latitude on any continent. So I looked up the latitude of Ann Arbor, and that's about where we're at, give or take 1, right? And so think about south of here in our continent, this phenomenon not even really being understood very well. So to address this, we started with some field sampling. I've talked to a lot of you today about field work and collecting specimens. We did this in the Southern Appalachians of North Carolina, collecting specimens of the mass shrew, Sorex araneus, which has an average mass of about 5 grams. We did this for about 2 years using pitfall trapping. We preserved all these specimens in our collection with a lot of other metadata and tissue samples and stuff. This is a nice sunny day on, I think, a winter sampling trip in North Carolina. If you don't know where I'm talking about, this is about where we're working here, where the black star is. If you don't know where Greensboro is, that's where I'm coming from, so that's about right there. Oops. And then we imaged these. So we imaged 125 Sorix scenarios on a Nikon machine, and to make a long story short, took a series of measurements on key parts of the skeleton. 4 craniodental measurements and 3 femur measurements. That's what a shrew looks like. Very gracile skeleton, very small animal. All the work is happening up here, kind of in the dentition. Otherwise, they're just kind of moving around under the leaf litter and finding invertebrates and things like that. So in all these plots I will show you, this is how they're laid out. So all the samples will be arranged by age class, starting kind of with the spring recruits. These things emerge from the nest and are thought to basically be kind of adult size, going through their first winter, and then basically making it to the end of their life. This is a nice study system because, unfortunately for them, they live a max of 2 years. And so we can track, cohorts, and they reproduce once a year, so we can kind of track the cohorts through space. All of these box plots will be labeled by color, by season. And then I just want to point out that some of you might be thinking this already. These are all cross-sectional samples. So we're going out and kind of sampling the traits of the populations through time, knowing a lot about the age categories and the demography. But to demonstrate true plasticity, you would need to go back and remeasure individuals through time. That's really hard with work like this. So this is kind of the best we can do, but we have a very good idea about kind of age structure of this population. Okay, so what did we find? So we took standard body mass measurements out in the field, and we found evidence for a Daynell's effect in Sorix scenarios. The largest individuals in the first year tend to be in the spring and summer, and then the smallest individuals are in the winter. So this is that winter shrinkage of the total body mass. And then you can see it's followed by this massive regrowth in spring. This is a time when individuals are regrowing their body mass. Both sexes are getting ready to reproduce early in the spring. And this is what the pattern looks like. This is the expected pattern, and not before shown kind of at our site. So we wanted another proxy of body mass. Sometimes it can be hard to measure shrews in the field. They get wet. They like to eat each other, so sometimes they don't have complete bodies when you get to them. So we measured a standard proxy for body mass in the skeleton, femur length. We took some other femur measurements as well, but these are just the femur length data. And to our surprise, this also exhibits a Daynell's phenomenon, with the longer femurs being in the first year summer, a winter shrinkage, and then this massive regrowth in the second spring. And then we took the standard brain case measurements as well. That also exhibits a Daynell's phenomenon. This turns out to maybe not be the best proxy for Daynell's, even though it has been the most common proxy, because as in many mammals, there's an ontogenetic flattening throughout the lifespan. And so there's this flattening trend happening, but there's still a Daynell's effect where there's a shrinkage and then smaller regrowth. All of the differences between the summer and the winter individuals were statistically significant, and I just put the percentages up here. So body size is shrinking on the order of like 13% between summer and winter, femur length by a little bit less, brain case height by about 11%. Okay, so we wanted to put this in the context of some other data, a very fragmentary literature, but a literature that has been accumulated over a lot of decades, actually. We wanted to know if the magnitude of plasticity we saw was actually something you could have predicted, so we did a meta-analysis. We found 75 studies on Sorix, population biology or physiology. And just to put a long story short, this encompassed 14 species spread across kind of the northern hemisphere, on 3 continents and 16 countries. And all these studies, our criteria were that they must include brain mass or brain case height as a trait. A few of them included both. the traits or the individuals need to be indexed by the age class and the season, and then it had to be on wild-caught individuals. This is just a geographic location of where all these studies came from. You can see it is highly biased towards Europe, and here we are here in North Carolina. So we just developed some bioclimatic proxies using WorldClim data. We use Bayesian linear regressions to just basically look at the magnitude of trait plasticity as a response of climate. And in these plots, what I'll show you, or our hypothesis, is that the amount of change, or the win or decrease, should be inversely correlated to the seasonality. So there should be or positively correlated. So there should be a greater decrease in places that are more seasonal, and less of this cyclic change or plasticity in places that are more aseasonal. And that's what we found. Here are the data for body mass, again from the set of 75 studies that had data for body mass. This is what the data looked like. Again, these are colored by the species group. The shapes represent the continent. You can see that it is still highly biased towards Europe, but there are some North American studies scattered. And this is what it looks like for brain case height. There's fewer studies that measured that, but still there's a strong trend as predicted from our work. So we also wanted to know, like I said, getting back to the reason we did this meta-analysis was what we found going out to our nearest field site and observing this phenomenon, what you would have predicted. So we did some posterior predictive modeling, and first we wanted to know if this result could have been predicted from the data. And we found that both body mass and brain case height were predicted from the data. They both fall well within the 90% posterior predictive intervals for those traits based on the data from all the other species. So this is our data here plotted with the red squares around it. What was also interesting was that we want to know if this phenomenon holds within and among species, right? It's an among species trend, but there's one species that's been investigated a lot, and again, that's the common shrew, Sorex araneus. So we did the same thing. We basically ran interspecific models without this species and tried to predict to the locations of the intraspecific data, and you can see that for both body mass and brain case height, the observed trend for araneus is falling within the 95% predictive intervals from the interspecific models in all but one case. This is the case that falls outside of the intervals. So, what have I told you? We're still convincing ourselves that this is all making sense. This is a crazy phenomenon. But this plasticity in body size appears to be a general phenomenon in this genus. And this genus is widespread, considered a high latitude specialist. Sorex araneus at our site in the Southern Appalachians undergoes about a 13% decrease in body weight and about an 11.5% decrease in brain case height in the winter, followed by a regrowth in spring. Our work provides, though, the first evidence of Daynell's below 43 degrees latitude, where we have both mass data and some skeletal data to go with it. And the first evidence actually from the appendicular skeleton. So we're kind of making our way along other parts of the appendicular skeleton to ask about this magnitude of plasticity. But this is absolutely wild. Those of you who think about femurs, whether you're paleo or developmental or anthro, I know you don't understand this either. And so we're trying to figure it all out with some histology and some other studies. The magnitude of plasticity, if you look across all the known observations, is related to climate. The models did okay. They predicted what we observed in Sorex araneus, and they also predicted intraspecific trends in the best studied species. And so there's a signal here that this is related to how seasonal the climate is. Okay. Let me move on to thinking about GI tracts, a nice soft tissue trait that's related to diet. Some of you think about diet or diet in extinct animals or diet in extant animals. And this work was funded by, or is funded by, a DRC grant from NSF, and this supported our work on GI tract plasticity in the Southern Appalachians, where we're still working on this project as well. So back to this model of rodent energetics. I remember that this is basically there's a lot of boxes here, there's a lot of details, but the gist is that energy balance is a kind of balance between food intake, maybe modulated by some environmental parameters, and these key reproductive processes and growth and maintenance of tissues, right? These are the things that individuals have at their disposal. These are the things they need to do with those. Really what's important here is what's the quality and the amount of the available calories and nutrients. And so it's not necessarily how much you eat, right? It's the quality of that food. I saw some of you eating cookies out here. I'm looking at you. And what sits between these things, right? What sits between the calories in your system to be used and their actual use? It's the digestive system, and specifically the GI tract and all its kind of components. And so back to this table of thinking about different organs and organ systems and how much they cost your body to maintain. The gastrointestinal tract is up here, and it's more even, right? In your body, it makes up maybe 2% of your average mass. It uses a little bit more than its mass-specific rate, right? It uses 10% maybe of your energy. In the rat model, it's actually more even. It makes about 5% of the mass, and it might use about 5% of the energy. So we actually don't know what to expect here, and we know that this structure is plastic. It can change in response to diet and climate. How it changes is what we were setting out to explore. So just a little bit of background on what on GI tract kind of biology, morphology. We're thinking about this as a really integrative functional trait. If you think about all the things the GI tract is doing for you, taking in food, optimizing absorption of some things, hopefully not absorbing toxins and rejecting those. It's a pretty integrative structure, right? And we're thinking about how it might contribute to seasonal adaptation, but also maybe things like community structure. It varies a lot across mammals. This is based on dietary guild. So as a rule, the longest and most complex GI tracts are in grazers and strict herbivores. The shortest and least complex are in carnivores, basically a tube. And this holds regardless of body size. So these herbivores, the deer and the rabbit, have relatively long GI tracts and complex tracts for their body mass. And these species, the weasel and the coyote, have relatively short ones for their body mass. And we know that it actually is plastic. So this is not new information that I'm giving you today. It can vary with diet. It can vary with energetic demand. So one of the major energetic demands in mammals is lactation. After that, after that, pregnancy. And so it can change in response to those energetic demands. And actually, we didn't have to do the meta-analysis here. Someone did it for us. We know that it's related to latitude, basically. So the degree of flexibility or the degree of plasticity is positively related to latitude. That's shown in this graph of small intestine length versus latitude. But this doesn't mean we're done, because these studies are still biased. So our knowledge of this phenomenon is also pretty uneven. 63% of the dots on this plot are studies of lab animals. And, well, not necessarily lab animals. They might be wild-caught individuals that then were maintained as colonies, but they're lab populations. Of the remaining 37% of these dots, 9 total species of wild mammals are represented. So we have good data on 9 species of wild mammals across the world for plasticity and GI tract length. So this is highly uneven. So we set out to work on this. Also in the Southern Appalachians, this time in a slightly different spot, in the Pisgah and Nantahala National Forest. We trapped for a year, sent one of my grad students up every month, heard a lot of stories about weather. The weather can be dicey, but she collected a minimum of 5 adult maniculatus. Sorry, I should introduce you to this. This is Paramiscus maniculatus. This is a North American deer mouse, a very widespread species in North America. A nice model because it's very abundant where we are at in trapping. So we're moving from shrews to rodents. So now we're moving to an omnivore model. It eats a variety of things. It can change its diet depending on what's in its environment. We took gross morphological measurements of the GI tracts of these animals we collected. These were length, wet mass, and dry mass of 4 sections of the GI tract. And then we analyzed those lengths and masses with respect to things like sex and breeding condition and their interaction. We also did some diet characterization with stable isotopes that we're trying to build on now with some other methods. I want to say that this work was headed up by my student, Olivia Chapman, who was a master's student with me, now a PhD student. I think she's holding her study species many years before she actually started the project. So maybe your fate is already decided, whether you know or not. And who is a University of Michigan alumni. I don't know if any of you ran into Olivia when she was here. I'll come back to Olivia in a minute. So this is what happens dietarily. We found that deer mice trophic levels change seasonally. In summer and in fall, these warmer colors, the diets of these individuals in this population are broader and of higher quality, likely incorporating things at higher trophic levels like fungi or arthropods. We can't parse that out with stable isotope data yet. And in winter and in fall, or in spring, early spring, there's a more restricted diet. And there's probably a diet of less, a lower trophic diet, probably less quality winter plants, really dry vegetation. So there is some dietary change happening. The relative GI tract lengths of the individuals caught in these seasons look like this. And the line is the model average from an additive model. And you can just see the magnitude of difference here. So again, these are winter individuals. There's a lot of variation, but this is the kind of average trend moving throughout the year. Oops. I don't know what that is. Can you guys read that? I hope it's true. There's about a 35% change in the length of the GI tract across these seasons. And so, again, this means that maybe, yeah. The GI tracts are longest in winter. These animals are increasing the amount of time it takes for poor quality food to move through so they can extract more nutrients from it. And so this is what we found. And we published this in the journal Mammalogy last year. The plasticity differs between the sexes, getting into, we have data at the level of the individual, so we can ask about how things like sex and breeding condition affect or mediate these patterns. There's some differences. This is length of the GI tract, its total wet mass, and its dry mass. There's really no sexual dimorphism in the GI tract, which I don't think you'd expect. What I do want to point out is, and the new results are a little bit nuanced, but what I do want to point out is the sex by repro interaction. What this means is that the sexes have different responses in their GI tract depending on whether if they're breeding or not. And what this means basically is that, here's the general trend, is that there's, at the population level, there's shorter GI tracts in the summer, access to better food. There's longer GI tracts in the winter to process poor quality food. But if you're a female and breeding, you don't have the luxury. Essentially, like your GI tract is long all year because when the spring and summer get here, you have the energetic demand of pregnancy and lactation. Right? So females, if they're breeding, tend to have kind of longer GI tracts all year. Okay, so what did I just tell you? GI tract plasticity seems to be a general phenomenon in herbivorous small mammals. Most of the species analyzed have shown some trend, depends on the seasonality of where they are coming from. These seasonal changes, in contrast to the shrew data, reflect the functional needs, right? And not metabolic rate of a tissue per se. There's really no way to kind of cut some corners with your GI tract is the way we're thinking about it, right? Because we actually need a longer GI tract in winter. So this change is kind of opposite of what I told you about the Dennell's effect. Peromyscus in the Southern Appalachians undergoes about a 35% increase in its GI tract length in the winter, followed by regrowth in spring. This is synchronous with some behavioral shifts to a poorer quality diet, and we're working to kind of refine that with some metabarcoding approaches. But this has some really exciting new synergies with the function of the GI tract, right? We're starting to think about how the microbiome might influence the amount of plasticity you need to undergo. Maybe a more efficient microbiome, more diverse microbiome kind of mediates the amount of plasticity that happens. We're working on intestinal microstructure because there's some differences in unit mass different sections of the gut, maybe some trade-offs in the gut, depending on what season it is and what section needs to work harder. And we're also thinking a little bit about toxin exposure and how leaky gut syndrome might kind of be induced or not by these changes. So these are all some, hopefully, future directions. Okay, I think I'm doing okay on time. You guys awake? 2/3 of the way through my classes, I try to get people to stand up and do something. You can move around, you can go to the bathroom. I want to talk now about how we're scaling this work up. So we've been doing this work thinking about individual level traits at a couple sites, but we want to know how this plays out across space and time, right? We want to know how life history properties and phenotypic properties of mammals are structured across space and whether they're changing through time. And again, this feeds into these ideas of having some trait data to inform policy and global kind of biodiversity management and conservation. So my lab thinks a lot about collections. I've talked to a lot of you today about collections. The collections really kind of provide the physical specimens that we can build on and extend knowledge from. And for our work specifically, they've been really great models because they're individual level data. So we have, for every data point, the spatial context, possibly the climate context, the sex, the life history data that we need to kind of have really rich understanding of why the traits vary. Museum specimens are extendable. So hopefully one of my students comes behind me and builds off of the knowledge we kind of layer on top of the specimens already to ask new questions. And these can be resurveyed through time, right? I can resurvey traits of mammals through time, whether all the mammals are in the collection or whether I go back to the collecting localities and resurvey in the same way. So these are really powerful models, right, for thinking about change through time. And this isn't about conservation only, right? This is also about ecology and evolution. Being an EEB department, you guys understand that this is valuable in its own right. And so the problem is that, as I alluded to in the start of my talk, if you go into a place like VertNet and look at all the specimen records and try to understand and extract trait data, about 10% or less of the records have some detectable, in this case, morphological reproductive traits. So this is a problem. Think about your favorite mammal collection if you work on mammals. Think about whatever your favorite taxon is. If 10% of the records had some trait data you could use, you'd hit a data wall pretty quick, and that's what happened with us. So we went out and looked for more funding to do some more digitization from specimens, and NSF awarded this award to us. It's called Ranges. It's a TCN, a thematic collections network. And the goals of Ranges are to do these things: digitize traits from about 1.2 million mammal specimens from all across Western North America, produce some 3D images for about maybe 3,000 to 4,000 of those specimens, basically extending the amount of traits and the number of traits we can get from them. Importantly, develop some trait data standards and some workflows for moving these trait data around, making sure they stay attached to the specimen records, but also become accessible to more researchers. The funnest part of this has been to reconnect a bunch of mammalogists and think about data together and think about next generation of trait data standards and really develop a community of practice around these traits that we're all collecting. Why Western North America? Well, it's a good starting point because it's the center of mammalian diversity and also endemism on the continent. And it faces a confluence of global change threats. You can pick your favorite: wildfire risk, drought, atmospheric river. Is that it? I mean, yeah. It is, yeah. Climate change, invasive species. So it's a nice place to think about having the baseline trait data for resurvey through time. The problem here is that a lot of the traits are analog. Mammalogists have been really doing a pretty good job, I'd say, through the years, with some exceptions, collecting trait data, right? They're just stuck on the tags. They're in an analog format. They can't be used by researchers very easily unless you go to the museums. In the cases where people have kind of done the hard work of digitizing, though, they're still not making it into these biodiversity aggregators. So like I said, if I went to the VertNet portal and tried to extract traits from the record set of mammals that are in there, about 10% of them would have some data I need. And this makes it really difficult to do anything with them, right? Whether your goal is research or policy or anything. So Ranges is a network of 22 institutions, including the Neon Biorepository, but a lot of great museums, some private, most university-based. Really happy to have Cody on board as a co-PI on this work. But this is kind of where all the institutions are at. And this is our geographic scope in yellow, so we've kind of sliced off the western half of the continent as a starting place. This has been fun, just as a side note, because this is a mix of collections, right? There's some very large representative mammal collections, historic collections. There's some small regional collections. But everyone's trying to think about these trait data, right? Getting them digital, getting them standardized. So what is Ranges going to do? I've talked about traits a lot. What specifically is going to happen? So data from specimen labels and possibly field notes and catalogs are going to become digital. This includes the 5 to 7 external measurements taken from mammals, 5 for most mammals, 7 for bats. I listed them all here, so tiny that you can't see them. But also reproductive data for females. This is things like breeding condition, pregnancy, embryo count, lactation status. Males, that tends to be things like whether they're scrotal or not, testis size, testis width and length. Those are kind of the raw data, the simple traits, getting back to this idea of like maybe doing some powerful things, some pretty simple traits. This is the trait data that mammalogists use. We're hoping to capture also some more broader contextual data, like ecological associations from field notes and field catalogs. And the goal here is just to extend the network of mammal specimens to basically go from the physical specimen. We have mostly the digital record available, but we want to layer these traits, make them digital and usable. I want to tell you really quickly about this because I know this is a big department with a lot of grad students and postdocs and even early career people. So we've been instituting this thing we're calling Imaging Mini Awards as part of Ranges. This is an opportunity not for us to go out and kind of slog through taxa and digitize taxa, but to have researchers do it for the stuff they're already working on to extend their own projects. So these are what are called Imaging Mini Awards. Who's eligible for these? The reason I'm telling you this is because most people in this room would be eligible if you're interested in doing something like this. Affiliated with the U.S. institution, be working on a trait-based project of some sort on a Western North American mammal species. We're giving priority to understudied or diverse clades, but really projects that can be integrative and layer new traits on top of the traits our project's already digitizing. So if you're interested in kind of functional morphology, but you're going to be able to use body mass that we digitize, and you want to collect some additional traits, this will be a good project for you, or a good opportunity for you. And our next funding round is in fall 2024. We'll try to announce it on X, Instagram, and maybe some Slack channels as well, but you should be able to find it on one of those places. If you're interested in this, please talk to me. I encourage you to apply. It's a chance for us to basically help the community digitize what the community wants to digitize and understand in terms of traits of mammals. So I will try to wrap up here. I want to come back to the idea of biodiversity data writ large. I described kind of the journey from primary observation. You know, my lab's fieldwork does a bit of this, collecting specimens, layering them with traits. But this is a long and arduous journey, right? From the collection to the digitization and the standardization and, you know, eventually the use for policy and management. Who's going to do this work? And how are we going to train them? I've talked to some of you about this today. I really, truly believe this. We have the best tools to do this of anyone at this institution, because we can take people out in the field and give them safe and fun field experiences. Some of them have never, some of my students have never been out in the field before. And so we have the opportunity to take people out in the field and see nature, see biodiversity. I also try to incorporate museum specimens in my work as much as I can. And these are really the two most powerful tools, right? Going out in the field and seeing specimens or collecting specimens to get people really engaged and have impact. But I try also to have my students think about the bigger picture, like I've talked about here today. How do we make these specimens digital? Basically, immediately after we come back from the field, how do we feed them out to other people to use? How do we layer them with traits that become digital as well? And that's a recipe that's pretty hard to beat, right? You ever watch this show? This guy is really hard to beat. So we have some great tools at our disposal. I want to end with this. I've got some evidence that this works. Some of you know Ben Hess out at the Museum of Zoology, who's the registrar. He used to be at the North Carolina Museum of Natural Sciences, and I worked with Ben at the North Carolina Museum of Natural Sciences back in the day. This is me circa 2009, sitting in the mammal collection, I guess not doing much work, but reading about mammals. And this was formative for me, right, to be in the collections, seeing the specimens, and doing the field work. And then I just want to come back to one of my current students, Olivia Chapman, who trained here at Michigan and did some time with Phil Myers in the field mammalogy class and worked with Cody a bit at the Museum of Zoology. And this is us touring a neon site in Virginia. And so there's some evidence this works, right? It's engaging people. It's opening up doors for people to explore nature on their own and do their own work. So I'm really excited to be part of this pathway of training people to generate and use these data. And so with that, I will just thank all my funding sources, collaborators on these and other projects, and my current and recent lab members. And I will be happy to take any questions.
Okay, do we have questions for Bryan? Here.
Does it? Yeah, you can hear me. I have a couple, and I'm going to try to formulate it as clean as possible for me. So I was very surprised about your body size and then brain size data, mostly because of, for instance, Bergman's rule, which suggests that in mammals or generally vertebrates, lower temperatures should support larger size. And so I guess my first question would be, why do you think it seems favorable to be small, like specifically? And then as a follow-up to that, because you said it's a plasticity, but if I remember correctly, you also mentioned that it wasn't like recapturing animals and measuring them. So it was like different specimens measured over time, right?
Yep.
So I guess my question is, you also said, use the term regrowth of the bones. Do you think like there's variable size of bones within the same individual, or is it rather that the frequency of given sizes in the population changes seasonally, which might be a result also of altered demography? And I can extend on that, but sorry, I don't want to throw too many questions at once.
Well, I'll start with the last one. We've tried to rack our brains about what demographic properties could be changing. And we can't really think of any that it would require us capturing, maybe having something like differential mortality happening. But then that bias would have to reverse in spring to then catch larger individuals in their second year. We've tried to think through it. This is a crazy phenomenon, right? And the more data we're collecting across the skeleton, we're asking ourselves harder and harder questions about bias and whether it's in there. But I don't know, do you have any ideas about what kind of demographic effects could be happening?
Yeah, so I remember one paper from Eric Pausma's lab on snow voles, I think, and they pointed out they had interesting counter variable selection. Essentially, what they were suggesting is, as the season starts, the animals start reproducing, and as the season goes, you get more juveniles. And then once you get the winter, substantial number of the juvenile dies off because they don't have enough resources, especially those that were born late in the season, because they're smaller, they have like fewer fat tissue, and there's less fat tissue, fewer energetic reserves. So it would be like, at the beginning of the season, you have only adults, then they start reproducing, you have increased number of juveniles, and then they die off during the winter. So the next winter, you stay with only the adults, and those juveniles that were born early, so they were already larger.
Yeah. This is, in a wild species, this is probably like the simplest we could get and control it. So they breed once per year in spring. So we don't think there's multiple cohorts coming in to the population throughout the year. And then I'm trying to get back to my slides, but it's going to be, close your eyes if you're sensitive to. Yeah, so this is really weird. These things are supposed to die basically after they reproduce in the summer, but in our 2 years of sampling, we caught two second year and second winter individuals, and they're showing the same trend. So these are all aged by their tooth wear, so we feel really good about their kind of what year they're in. So I totally get it. We've tried to this is a really simple system, so that helps. We're trying to track when they reproduce to mitigate bias, but yeah, it's we're trying to figure it all out. Yeah. But anyway, basically, the idea is they're kind of growing really quickly, and when they come out of the nest in the spring, they're kind of adult size, and there's not another reproductive event till the next year.
All right. Thank you so much, and also, if I didn't mention it at the beginning, great talk, very interesting research.
Thanks.
Thank you.
Hi, yeah, super cool system. I was wondering about the gut plasticity, and then you were saying the microbiome. I was wondering if you have any ideas or theories on how the microbiome, community structure or dynamics plays into the plasticity side. I can imagine just different species metabolizing different things, based on food quality, but then more in terms of the link between the size of the gut itself, right?
Yeah, I mean, I think building on what we have figured out already, our first hypothesis is that maybe, our first test is that maybe with a more efficient microbiome, so things that can digest fibrous foods better or even toxic foods, might mitigate the amount of plasticity that you have to experience. So I guess in this system where we're kind of working with cross-sectional samples, it would be asking whether, every individual relative to the seasonal mean, basically taking those residuals, will be correlated with something like microbial diversity or microbial functional diversity. Does that make sense?
Yeah.
I mean, I'm not a microbiologist, and so I told some of you earlier I'm trying to resist even doing this project. But so I'm happy to talk more about it. But that'd be my first hypothesis. That'd be my first question, is whether the amount of plasticity that individuals are having to kind of undergo could be mitigated by how healthy or diverse their microbiome is, or functionally diverse.
Yeah, I had another, thing to throw out about this. I was wondering if foraging effort changes throughout the seasons, and whether if you're a lower condition individual, whether you need to be more active in the winter just to stay alive, and so there might be just maybe they're out there more, getting caught more, and then while the higher quality or higher condition individuals might be less. I don't know if you know about foraging effort.
No, I don't. And I mean, it's a good question, and there's lots of scenarios that one could think of to explain this, and we want to explore them all, but we don't have those data. But this is actually maybe the most parsimonious data based on the literature that exists, right? To kind of have a demographic factor happen basically across space and the span of almost a century and many different populations would, I guess, be totally unexpected. So we're kind of building on the knowledge that exists and assuming that's kind of the population demography of our site. But it's a good question. We don't know anything about how individuals kind of differentially forage or die at different seasons.
Yeah. Well, I'm going to follow up with more questions about this phenomenon. I'm curious, you remind us, what was the latitudinal range of your spatial samples here? You said something about
in the meta-analysis.
Yeah. It was something that I remember 40.
Yeah, this was.
So your high latitude to 43 degrees, was that it? Something?
No,
like that?
The 43 degrees was saying below 43 degrees on any continent, there's no data, data on mass and skeletal proportions.
Oh.
So some of these populations over here, or I don't know, maybe here have just body mass, which is not a great trait on its own.
Well, I get my question was, do you know what aspects of seasonality are predicting these changes? Because you can have seasonality. There's certainly seasonality of temperature, and that's one that is often implied when that word is used. But there's seasonality of day length. There's seasonality of precipitation, and lots of other things. And I'm wondering, is it one thing? Is it many things? Have you analyzed the climatic variables in such a way that you can see what combination, or even whether those variables are capturing what you need?
Yeah, I wish I had that in here. So instead of going one by one with the climate variables and trying to find the best one, we just assume they are correlated, which they are correlated over this region, and we just PCA transform them. So basically the climate axis is PC1 of that ordination, and the variables that load most strongly on that are the seasonal temperature and precip variables. So it's like temperature of the driest quarter. It tend to be like the seasonal variable. PC1.
PC2 is frequently seasonality. It's the contrasts between the seasons instead of the general trend through them.
Yeah, I wish I had the biplot here, but basically the variables that load most strongly on the PCA of these sites are the seasonal temp and precip.
I'm wondering if you were to parse the data out in a different way, whether you would find that the change in body size is proportional to the degree of seasonality of the sites, because there's going to be big changes in the amount of seasonality from low to high latitudes or from drier to wetter places, and the climatic data should capture that.
So, I mean, thinking about this, if I understood your question right, that's kind of what this shows, I think. But it's complicated, right? So you can think of this as like seasonality, increasing seasonality. And this is temperature and precip of a couple quarters of the year, predicting, the amount of decrease that happens in the winter. And some of you know these taxa. Some of these are Asian or North American, that are the teal ones that are kind of falling more off the map. We want to go kind of back to these models and ask, well, what are the regions of the world where the shrews might be right on the edge, right? So there's been some studies from coastal Spain that maybe the phenomenon doesn't even occur there, or it's like very minor. Maybe some places on the West Coast, North America. We want to kind of ask how that might, how that will correlate with local conditions.
Okay, I think we'll stop questions there. If anybody would like to chat with Bryan here as we end, feel free to come up front. There's going to be a few of us go to Red Hawk if you'd like to take your questions to the brewery or the bar. So, alrighty, thank you.